Hello, good morning, friends, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here with the lecture The Basic Structure of All Living Creatures. So, friends, this is very important topic for all the life sciences exams such as GRF, SRF, and ARS, also for the CSI. In this lecture, we will discuss about the different concepts or mechanisms of cell biology, and the content of our today lecture is we will discuss about the origin of life and origin of eukaryotes. Later on, we will discuss about the difference between the prokaryotes and eukaryotic cells. Later on, we will discuss about the structure and function of cell membrane and cell wall. And at the end of this lecture, we will discuss about the cell division. So, friends, let's start our topic with the origin of life. So, you know that origin of life is a very complex process, and the evolution of life is a continuous process. Also, there are irreversible changes, and the earlier organism was very simple in nature. They have very simple structure, and the modern eukaryotes have complex structure like archaebacteria, and in the complex plants and animals, they are higher organisms. Okay, friends. Now come to the universe and origin of Earth. So you know that our universe has very old history, and approximately 13.79 billions of years it is originated. And from that time, it is continuously expanding. This is the unique property of our universe, that it is continuously expanding. And the expansion of the universe can be understood by the Big Bang theory. So, what this theory says, according to this theory, thus at a single point, our universe increase in the energy, biomass, and space. And these things change in these three particulates was for the expansion of universe. Likewise, there is a one process which cause for the origin of different planets, galaxies, and stars. Okay, friends. So what is the assertion? In the assertion, at pro-planetary days, there will be a accumulation of more gases. This particular point it attract more gases by gravitational forces and cause for the origin of different planets, galaxies, and stars. And our Earth is also originated by this process. Nearly 4.54 billion years. Okay, friends. Now come to early Earth atmosphere. So, friends, you know that our Earth is originated from the solar nebula, which have very high temperature. So, earlier in our Earth, the temperature which is very high, and it is like a molten magma. Okay, friends, and there is no oxygen, no water. So, the atmosphere, the earlier condition is very hardy. And with the time, this molten magma is cooled down, and form the solid crust. This is called Earth's surface. Okay. Now this Earth's surface are able to hold liquid like water. Okay. Also, in the earlier Earth atmosphere, there are volcanic activity, high volcanic activity, and the asteroids are also very common. So, one of asteroids which found very far from the sun, which act as a water delivery system on this planet. Okay, friend. So this is the carbonaceous chondrites, which is the source of water in this planet. Okay. Now, the you know that first organism is the archaebacteria, which is heterotrophic in nature. And generally, it is assumed that these organisms are evolved in the hardest place of the earth. Like it could be the bottom of the sea. Bottom of the sea. So first life is originated in the bottom of the sea. Okay. And the first life when it is originated, it is approximately 3.7 billion years ago. Okay, friends. Now, some studies also found that in the earlier history there are zircon rocks. In these rocks they found carbon material. And you know that carbon material is the main component of organism. So maybe possible that life is originated 4.1 billion years ago. Okay, friends. And the oldest known fossils is the stromatolite. Stromatolite. Okay, fine. This is known as oldest fossil, which is 3.7-year-old. Now, the first experiment, first theory, that is the abiogenesis theory, which states that the present life is originated from the abiogenesis. So, what is abiogenesis? Abiogenesis means the biotic components are formed by the abiotic factors, abiotic components. So, what are the abiotic components in earlier Earth? They are methane, ammonia, H2, N2, H2O. These are the earlier component of Earth. Okay. So in 19, this theory given by the abiogenesis theory given by the Operon and Eldon. Independently, they proposed this theory in 1924 and 1929. Okay, friends. And this theory was proved by the experiment conducted by Miller and Urey in 1952. So they take these compounds under high light and electric potential, and they found that there will be a formation of amino acids, and this is they called as a prebiotic soup. Prebiotic soup. So this prebiotic soup is responsible for life on this planet. Also, some researchers suggested that in the earlier Earth, there will be a RNA world. Means DNA is not there. So RNA is able to all the work, like it act as an enzyme, it act as a protein, and also act as a additive material. Also, some new studies suggested that the life is originated near the nuclear reservoirs, because in the earlier Earth there are lot of nucleoactive compounds like radioactive compounds like uranium, radon, and which have very unstable. They are very unstable. So they create very high temperature and they bound these compounds into the organic form. Organic form. Also, there are one latest theory. They propose that the life is not originated in our Earth. It is seeded by the comets and meteors from different planets or another planets. And this is proved that some asteroids which are coming from other planets having clay minerals, and also they have amino acids. Okay, friends. So there are certain some theories, and also the water on almost 4.31 billion years ago, and the eukaryotes comes at approximately 3.7 billion years ago, and the first oxygenic bacteria, that is cyanobacteria, comes 2.3 to 2.4 billion years ago. Okay, friends. So this is the history and origin of life on this planet. Now come to the cell. History of cell biology. So the cell is the basic structure and functional unit of life form. The cell comes from the Latin word cella. Sometimes it can be asked in exams. And the meaning of this cella is the small room and chamber. In 1665, Robert Hooke discovered the cells in a cork. So friends, cork is a dead tissue. Cork is a dead tissue. So the cell discovered by the Robert Hooke are the dead cells. And also the term soil is coined by the Robert Hooke. And the study was published in the book Micrographia. The Micrographia and this book is published by the Royal Society, London. And the title of full book is the Micrographia, some physiological description of minute bodies made by magnifying glasses. So this is the full title of the book. Okay, friends. Later on, the scientist Anton von Leeuwenhoek discovered the living cell in algae spirogyra in 1674. So if someone asks that who discovered the cell, then answer goes to Robert Hooke. And who discovered the living cells, first living cell, then answer goes to the Anton von Leeuwenhoek. Later on, 1839, Theodor P. Schwann and M.J. Sliden proposed the cell theory. And they proposed that cells are a common unit of structure and development. And later on, this theory was expanded by the Rudolf Virchow, who stated that new cells come from the pre-existing cells by cell division process. Later on, in 1859, Louis Pasteur found that life form can occur spontaneously. Later on, in 1981, Lynn Margulis published the endosymbiotic theory. We will discuss about this theory. So these are some important contribution of researchers in the field of cell biology. Okay, friends. Now come to the modern cell theory. So according to this theory, all organisms are composed of one and more cells, like eukaryotic cells have many cells, and all cells come from pre-existing cells, proposed by the Rudolf Virchow. Also, cell have the hereditary information in the form of RNA and DNA, and necessary for regulating cell functions and for transmitting information to the next generation of cells. Likely, a similar species organisms have similar chemical composition because their function is also similar. Like also energy flow occurs within the cells, means there are certain metabolism activity, the cationic broken out to complex compounds, and anionic like synthesis of complex compounds. Then last is the activity of organism depend on the total activity of independent cells. Means two and more cells comes together for controlling a functions, which regulate the total activity of organisms. So this is the modern cell theory. Now come to prokaryotes and eukaryotic cells. So friends, this is the structure of a bacterial cells, and you can see the structure of bacterial cells. There are cell wall and plasma membrane is the uh, common component. However, there are certain difference that cell wall of these prokaryotes are made of polysaccharides and muramic acid. Okay. Then plasma membrane, then nucleoids. They have there are no true nucleus in the prokaryote cells. Instead of nucleus, they have nucleoid-like structure in which this structure is stay in the cytoplasm. Then ribosomes. So prokaryote cells have 70 S type of ribosome, while the eukaryotes have 18 S type of ribosomes. Okay. Other than this, bacterial cells have plasmid, that is extra chromosomal DNA, and this is used for the antibiotic resistance. Okay, friends. Also, they have flagellum and pilus structure. They are the outgrowth of cell membrane and used for the locomotion or movement and communication. Okay, friends. Also, they have one capsule layer outer the cell membranes, and this structure have protect against viruses. Okay, this structure protected against viruses. So this is the common structure of prokaryote cells. And what happened to the plant cells? In plant cells, they are true nucleus is present. Then there are membrane-bound organelles like chloroplast, endoplasmic reticulum, Golgi vesicles are present. Also, there are large central vacuoles are present, which work for storage, act as a storage house of cells. And there are amyloplast is there. Then cell membrane and cell wall is common in both, and cytoplasm and ribosome. So ribosomes are the 18S in case of plant cells or eukaryotes. Okay, and rough endoplasmic reticulum. The ribosome present on rough endoplasmic reticulum and used for protein synthesis, while smooth endoplasmic reticulum don't have ribosomes and they use for lipid synthesis. Okay, friends, these are the common difference between the prokaryotes and eukaryotic structure. Now, this question can be asked in exams that what are the difference between the prokaryotes and eukaryote cells. So, first is the complexity. So, you know that prokaryote cells are very simpler and unicellular, while the eukaryote cells are complex in nature and they have multicellular, more than one cells. Now come to size. The prokaryote cells are very small, near to 0.5 to 2 micrometer, while the eukaryote cells have very large, it is between 10 to 100 micrometer. And DNA in prokaryote cells, they have single stranded circular DNA, while in case of eukaryotes, it is double helix, and most common is the double helix model given by the Watson and Crick in 1953. Okay, friends. Then membrane mode only already discussed that they are absent like Golgi body, endoplasmic reticulum, and nucleus are absent in prokaryote cells but present in our eukaryote cells. Nucleus, their true nucleus is absent. Instead of this, nucleoid structures are present. And in eukaryote cells, developed nucleus is present, which have your nuclear core, nuclear envelope, and all the structures. Ribosomes in prokaryote cells, they are 70S ribosomes, while in case of eukaryotes, there are 18S ribosomes. What about plasma membrane and cytoplasm? They are both present in prokaryotes and eukaryote cells. Also, the growth rate of prokaryote cells is faster and eukaryote cells is slower. The curve of growth is, in case of bacteria life, it is J-shape, and in eukaryotes, it is sigmoidal curve. Okay, so this is slow and this is, there are certain phases like lag phase, log phase, stationary phase, declining phase. But here only exponential and then chromosomes. They have one long single loops of DNA and plasmids, while in case of eukaryotes, there are more than one chromosomes. Receptor of plasma membrane generally absent but present in case of eukaryotic cells. Now talk about genomes. So prokaryote cells have haploid genome, and these eukaryote cells have diploid, and there is large amount of non-coding and repetitive. This is called as a junk DNA, not code for any proteins. And plasmid is very common in prokaryote cells, while it is not common in eukaryotic cells. Introns, they are absent in prokaryote cells, present in eukaryote cells. Glycocalyx layer generally have, and eukaryote cells don't have this glycocalyx, but if there is cell wall is absent, then there will be a glycocalyx layer. Cell cycle is very short. It is near 20 to 60 minutes, but it is long in case of, but in the case of eukaryotic cells, it is very long, near to 12 to 24 hours. And cytoplasmic streaming, there is no cytoplasmic streaming in the case of prokaryote cells, but present in the case of eukaryotic cells, means in which, so cytoplasmic streaming is present in the case of eukaryotic cells. So what is the meaning of cytoplasmic streaming? Means your cytoskeleton of eukaryote cells create a force which cause for the inward flow of cytoplasm, and it is useful for nutrients mobilization in the cells. Nutrients mobilization. Okay, friends. Then lysosomes and peroxisomes are absent in the case of prokaryote cells and present in eukaryotic cells. Transcription and translation process simultaneously occurs in prokaryotic cells, but in case of eukaryotic cells, it is occurs in nucleus. Transcription occurs in nucleus, while translation, synthesis of proteins take place in the cytosol. Transcription is the synthesis of RNA from DNA. Reproduction, generally the asexual reproduction is present in prokaryotic cells, but in eukaryotic cells there are sexual reproduction, and the prokaryotic cells are reproduced through binary fission and also budding. But in eukaryotic cells, mitosis and meiosis are the types of reproduction. Stone proteins, these are not present in the prokaryotic cells but present in the eukaryotic cells and bound to the DNA. And they are resin, arginine and histidine. Flagellum, it is simple in the case of eukaryotic cells. In eukaryotic cells, generally not present, but if present, it is complex and built from the microtubules. Cytoskeleton, late, there is no cytoskeleton in the prokaryotic cells, but in eukaryotic cells, it is made up of active and microtubules. So friends, now come to origin of eukaryotic cells. So you know that earlier there are occurrence of prokaryotic cells, but how the eukaryotic cells are evolved from these prokaryotic cells. So in the first process, the prokaryotic cells start the unfolding of plasma membranes, and they give rise to the endomembrane component like nucleus and endoplasmic reticulum. Means their membranes is bound to the, they have projections like that and bound to the your center and from the nucleus and endoplasmic reticulum. Now, in the first endosymbiotic events, the ancestral eukaryotes, like this aerobic bacterium, consumed by this prokaryotes. So this consumed this aerobic bacteria and developed into the heterotrophic eukaryotes. Okay, friends. And the second embiotic events, the early eukaryotes consumed photosynthetic bacteria and evolved into the, this photosynthetic bacterium evolved into the chloroplast, and like this our modern photosynthetic eukaryotes are evolved. So in the history there are two endosymbiotic events occurs. In first events, the prokaryote cell engulfed the aerobic bacterium and it converted into the present time of mitochondria. And second, endosymbiotic, they consume this photosynthetic bacterium and convert into the your chloroplast. So like this, the over present eukaryotes are developed. Now come to the cell wall. So friends, plant cells are surrounded by a mechanically strong cell wall. This cell wall provides the strength to the cells. And this thin layer consists of scaffold of cellulose fibers. So cellular fibers embedded in a matrix of polysaccharides. So matrix is made up of polysaccharides. There are certain polysaccharides present in the cell wall, and in which these fibers are embedded, and they form a strong network which provide the strength. Okay, how they assembled by the covalent and non-covalent bond. And the matrix also contain, this polysaccharide matrix also contain some structural protein, enzymes, phenolic polymers, and other materials that modify the whole physical and chemical characteristics. And this is the common structure of cell wall. So in this, this is the cellulose microfibrils. Okay, and this is polysaccharide that hemicellulose, in which the soluble proteins are present and pectins. So this form the cell. And this is the composition of cell wall. So microfibrils made of cellulose, and cellulose is a polymer of beta D14 glycan, 14 beta D glycans. Second, pectins. It is made of, there are which pectins are present: homogalacturonine, rhamnogalacturelatron with arabinon galactons, and this pectin form the hydrated gels, in which your hemicellulose network embedded, and it forms a hydrophilic pillar to prevent aggregates and collapse the network. So what is hydrophilic and hydrophobic? Hydrophilic means they are water loving. And what is the meaning of hydrophobic? They are water repellent. Okay. Then hemicellulose, xylo, glucone, xylene, glucomanone, arabinoxylene, cellulose, beta-D-glycan. These are present in the cell wall, and this bind to the surface of cellulose and microfibril together. So it form the cohesive network. Okay, friend. Then come to lignin. So they are H-lignin, P-carbonyl alcohols, coniferyl alcohol, G-lignin, sinophyl H-lignin, biphenyl ether, and cinnamyl alcohols. So these types of lignin is present in the cell wall. And structural proteins, HRGP, hydroxyproline-rich glycoproteins, which are 55%, and PRP, proline-rich protein, is 0 to 20%, and glycine-rich proteins. So the primary cell walls is contain 25% cellulose, 30% hemicellulose, 40% proteins, and 2.5% of proteins. While the secondary walls are contain mostly 90% of cellulose, like in cotton fibers. So this is the composition of cell wall. Now what are the important functions of cell wall? So it regulates the cell volume, it determines the cell shape, means it stops the expansion of cells. Also it acts as a barrier for different pathogens. Also it protects the protoplast, and it provides the mechanical strength and allow plant to grow up. Then it also regulates the plant morphogenesis. Also it maintains water relation and transmission flow because it provides strength to the xylem. Okay. Then it is the cell. This cell wall or cellulose can be used for the production of paper and textile industry. So friends, these are the function of cell walls. Now we come to cell membranes. So you know that all living cells are covered by a thin, this is very thin layer, delicate, elastic, and the important feature is that it is selectively permeable or differential selectivity. Means cell membrane are not allowed to move all the ions into the cells. Means they are differentially selectivity. So this is also called as a cell membrane, plasma membrane, biomembrane, or plasma lemma. So there are certain models are proposed for the cell membrane structure. First model was the sandwich or trilamellar model, which was proposed by the Davison and Danieli in 1935, which was later unaccepted. Later on, the second model is unit membrane model by the Robertson in 1951-9. It is also rejected. Why this model are rejected? Because they are not able to describe that how the cell functions, how the cell function. Okay. Then the latest model proposed by the that is called fluid mosaic model. This is the most accepted model given by the Singer and Nicholson in 1972, and this is the widely accepted model. According to this model, all biological membranes have a similar basic molecular organization, which are also proposed during the cell theory, and they consist of a bilayer of either phospholipid or, in the case of chloroplast, glycosyl glyceride as a fatty acid in which protein embedded each layer called as a bilayer. There are two layers of the this fatty acid in which protein are embedded. Okay. Then we will discuss about the phospholipids. So class of lipids that contain two fatty acids. are covalently linked to the phosphate group and form the head group, such as serine, choline, glycerol, and inositol. So these fatty acids form the head group of this layer. Okay. And second one, the non-polar hydrocarbon chains of fatty acid, which form a resin that is exclusively hydrophobic. That is, so it made hydrophobic layer. And the head group is the highly polar and hydrophilic nature. So we can say that this layer of fatty acid shows the both property, amphipathic property means the head group is the your hydrophilic and your fatty acid chains. This non-polar hydrocarbon chains is the hydrophobic properties. Also, these phospholipids are dispersed asymmetrically across the plasma membrane, and this gives the membrane sideness. Shape of the cell is given by the membrane sideness. The outside leaflet of the plasma membrane, which faces outside the cell, is the different from the inside of the cells. So the two part of the cell totally different. The fatty acid chain of phospholipid and glycosyl glyceride present in the chloride this here. It is 14 to 20 carbon bonds. If it is linked by the single bond, then it is called as a saturated fatty acids. And if double and triple bonds are present, then it is called as a unsaturated fatty acids. The double bond present in the unsaturated fatty acids can rotate, so create a kink in the fatty acid chain. They prevent the high packing of phospholipid in the bilayer, or it provides the sideness of the cell. It provides the fluidity of membrane, which is critical for the numerous membrane functions. So friends, this is the structure and arrangement of the phospholipid. And now come to proteins. So friends, there are three types of protein which are associated with the cell membranes and associated with the lipid bilayer. So first is the integral proteins. So these proteins are implanted in the lipid bilayer and they span the entire width of phospholipid bilayers. Means they are connected to the both phospholipid bilayers. And one part of this protein interact with the outside of cell, and second part interact with the hydrophobic core of the membrane, and third part interact with the interior of the cell, means cytosol. And these protein always act as a ion channels. Okay, and there are certain receptors that participate in the signal transduction. So integral protein है, and second one is the peripheral protein, and these are bound to the membrane surface by a monovalent bond that may be ionic or hydrogen bond. Then that can be dissociated from the membrane with the high salt concentration or geotropic agents, which break this ionic and hydrogen bonds. And this peripheral protein, surface protein, interact with the plasma membrane and component of the cytoskeleton, such as microtubules and microfilaments. And the third type protein is the N-core protein. And these protein bound to the membrane surface via the lipid molecules, and it is covalently attached. And these lipids includes malic acid and palmitic acid because they are attached with the lipid molecules. Like GPI N-core proteins, glycosyl, phosphatidylate, inositol, N-core protein. Like these are the N-core protein. So these lipid N-cores make two sides of membrane, fatty acid and perennial N-core occurring on the cytoplasm facing, and second one is the GPI N-core, which is the outside of the cells. And this is the common structure of cell membrane, which we discussed earlier, that it have phospholipid bilayer and it made a hydrophilic head made up of polar like serine. These are the fatty acids which made this. Then hydrophobic traits, then peripheral proteins, integral proteins, glycolipids, protein channels which transported the proteins. So this is the common structure of cell membrane. And what are the functions of cell membranes? So cell membranes forms outer boundary. It also separates the cytoplasm. It is selective permeable, the movement of ions and other molecules, and accumulation of ion molecules maintain cell turgor, means it maintains the cell turgidity, site for enzymatic reactions, cell adhesions and ion conductivity, and for signaling process, and it also acts as a border of cell organelle. So friends, these are the function of cell membrane. Now come to cell division. So friends, cell division is the process by which a parent cells divides into two daughter cells. So this is the parent cells and it is divided into two cells. So this is the type of prokaryotic cells, prokaryotic cell division, and prokaryotic cells how they are divided. Cell division is mostly binary division. Okay friends, some prokaryotes are divided by the buddings. So in this what happened? This is the process of binary fission. So replication of this circular prokaryotic chromosomal began at the origin of replication. So this is the origin of replication, where the origin of replication process started and continues in both directions at once. Okay friends. Then the cells began to elongate. These cells are started elongated, and there is one protein that is FTS proteins migrates toward the midbody of the cells. So these are the FTS proteins which migrate towards the center of the cells. So their DNA replications occurs. So this duplicate chromosome separates. Now the separation process starts and continue to move away. Now they are moving away from each other toward the opposite end of the cells, means they are moving the opposite sides of the, and these FTH protein form a ring around the periphery of the midpoint between the chromosomes. So this make a periphery of this, and this accumulate in the center of cells. The FDZ ring direct the formation of a septum. There is a formation of septum that divide the cells, and plasma membrane and cell wall material accumulated. After the septum is completed, the cell pinch into forming two daughter cells. FTSZ is dispersed through the cytoplasm in the new cells. So this is the common types of cell division found in the prokaryotes, in which the first replication starts, then this FTZ protein move towards this center, and these two chromosomes, duplicate chromosomes, moving in the opposite direction. Then this FTS Z protein accumulates in the centers, and it forms the septum, and the cells are divided into the two daughter cells. Now come to the eukaryotic cell division. So eukaryotic cell division are more complicated than the prokaryotes. And in the eukaryotic cells, there are two types of division. One is mitosis and second one is the meiosis. So mitosis generally called as a vegetative division, while meiosis is the reproductive division. And it produces daughter cells which are genetically identical to the parent cells like this. And this meiosis produces the haploid gametes for the sexual reproduction, because it is generally present in the sexual cells like pollen, anther. And meiosis is the form of eukaryotic cell division that produce haploid sex cells or gametes from diploid cells. The process takes from one DNA replications followed by two successive karyokinesis and cytokinesis. Okay, friends. So we'll discuss in detail about the mitosis. So mitosis, this is the interphase stage, and this is the phase of mitosis in which there are some certain phases like interphase. This is also called as a resting phase. And in this phase, the cell is prepared for the DNA replication. Then some cells there are G1, G0 stage. This is also called as a quiescence phase, in which cells are metabolically active but there is no division take place. Later on, the first stage is the gap phase, gap 1. So in this phase, the cells are prepared for the DNA synthesis, and preparation of DNA synthesis takes place first growth phase, and it is also post mitotic gap phase, and the duration is very high. This is from 6 to 12 hours, and all types of RNA and protein are synthesized during this stage. And later on, the second stage is the synthesis phase, which is also called as a S phase. And the DNA replication takes place during this phase, and the formation of histone and duplication occurs in this phase. Then third one is the G2 phase. This is a small time. It takes a small time, like 3 to 4 hours. And what happens in G2 phase? It is the pre-mitotic gap and preparation for mitosis, stop DNA synthesis, and continue RNA and protein synthesis, and organelle and spindle formation take place during the G2 phase. Then there are some checkpoints which regulates the activity of, means accurate activity of this process. So G1S, so one checkpoint is present here, G1S, to check the appropriate cell size and DNA damage. If there is any DNA damage occur, then there will be activation of DNA repairing system. Okay. Then second one is the G2 checkpoint, and at the end of check DNA, it check the DNA damage. Okay, friends. So this is the now the mitotic phase, also called as a M phase. And this mitotic is the equational division. Start with the nuclear division, consequently to the karyokinesis, means division of the nucleus, then separation of the daughter cells, and end with the cytokinesis, means division in the cytoplasm division. So first phase is the prophase phase. This is the first stage of the mitosis. And in this phase, chromosomes are shorter and thicker, and sister chromatids link to the centromeres. Now the centrioles moving apart opposite directions and reach to the opposite poles. So this activity occurs in the prophase phase. And what occurs in the metaphase phase? In this phase, each chromosome get attached to the spindle fibers by its centromere, and although chromatids are still connected to the spindle fibers, and chromosomes are lined up in the metaphase plates like that. And metaphase checkpoint, there is one checkpoint which check for chromosome, that chromosomes are attached with spindle fibers or not. If there is not, then there will be a repair. Then anaphase. In this centromere attaching of two chromatids now start divides, and two sister chromatids of each chromosome separates. So now formation of sister chromatid started, and they are drawn apart towards opposite poles. They are moving to the opposite poles by the shortening of the spindle fibers. And the last phase is the telophase, in which two sets of daughter chromosomes reach to the opposite poles, and now spindle fibers get vanished, and the nuclear envelope is created around the chromosomes cluster, and nucleus, Golgi complex, and ER are formed. Now, after the karyokinesis process, cytokinesis process takes place. Generally, in plant cells, cell wall formation begins with in the center with the formation of cell plates. This grow outward to meet the lateral walls. Thus, cytoplasm is split into the two parts, and this cell plate becomes the central lamellae. Okay, friends. So this is the mitosis process. So what happens in mitosis process? So mitosis process, it starts with the interphase. Then interphase, what are the phase? There will be a G0 phase in quiescent. Then as G1 phase in which DNA prepare for DNA synthesis. S phase where DNA replication takes place. G2 phase in which the organelle and spindle formation take place. And G2 phase in which this is the end phase of this mitotic. Then in mitotic phase, there will be a prophase, metaphase, telophase, and anaphase. Prophase, metaphase, anaphase, and telophase. So prophase, in which sister chromatid linked to the centromere. In metaphase, they are lined up in a equators. Anaphase, there will be a formation of two sister chromatids, and still they are attaching and moving the opposite pole. And telophase, they reach to the end opposite poles, and now spindle fibers are get vanished, and there will be a formation of nucleus Golgi complex. Then after the karyokinesis, there will be a cytokinesis which form the two cells, daughter cells. Now come to the meiosis. So friends, interphase is similar to this mitosis phase, and in this meiosis there will be a formation of gametes, sperm, and egg cells. So in the meiosis, there will be two meiosis, meiosis first and meiosis second. So meiosis second is similar to the mitosis. Okay, but there will be some changes in meiosis phase. Meiosis first, there will be in prophase phase. Interphase is similar, then mitotic phase, meiotic phase. The first stage is the prophase phase. In prophase, there will be a disintegration of the nuclear membrane. So nuclear membrane got disintegrated, condensed chromosome, and cystochromatid attached to the centromere. Now the homologous chromosomes pair up. So it forms the tetrads or chiasma. So there are some sub-stages in the prophase stage like leptotin, zygotin, pachytin, and diplotin. So leptotin phase, there will be a initiation of recombination, means there will be recombination initiated. Then formation of bucket stage, it is the zygotin also called as a bucket stage, in which these threads are paired, and formation of synaptonemal complexes like that. Next, the pachytin, in which recombination and crossing over take place between this homologous chromosomes. And diplotin, there is formation of two threads, and tetrad are visuals. The second phase is the metaphase. It is like that, in which these tetrads are lined up in the metaphase plates. Anaphase, these tetrad are visual and move toward the opposite poles. Then telophase, same as nuclear membrane reappears and separate the product of meiosis first. Then DNA relax and go to the DNA, this meiosis second, in which DNA condensed. Then in metaphase second, they are lined up, and anaphase the centromere splits and sister chromatids are pulled to the opposite poles, and nuclear membrane reforms, and there will be this interkinesis. So like that, this different cells are found in the meiosis process. So friends, this is the some description about the cell biology, and in next lecture we will cover about the function of cell organelle. So thank you very much. Thank you very much.